So now we've reached the end of a journey. We've been trying to get to grips with this remarkable structure, to address the question of why it is that two completely separate DNA molecules, provided they have the correct complementary sequences, are able to wrap around one another to form this stable double helical structure, which is essential for the DNA's biological role. And what we've seen is that you can't address that question by assuming that it's just something that biology does, that biology sort of stitches it together, because it will happen quite spontaneously in the absence of any cellular apparatus. And we've seen also that you can't just look at the intermolecular forces within the double helix and say, oh yeah, that's what holds it together. Because this is about a chemical equilibrium. So we have to think also about the alternative form of DNA in which the double helix falls apart and you have two completely separate molecules with no fixed structure. And you have to consider all the things that change during the assembly process that leads to the double helix. So what we're going to do here is just briefly to review the key features that emerged as we explored this really quite complicated story. So this means that we can't base this discussion just on asking the question, how strong are the bonds that hold the structure together? Because we have to compare the alternative interactions that would be formed with the DNA's surroundings, which means the water molecules in which it's dissolved, if it were to unfold. But more than that, we also learned that it isn't just about the strength of those interactions either, because there was this other factor, the entropy, that came into play and was also a really important determinant of what happened. So this equation is going to be the key one for this discussion. So delta G is called the free energy change, and what we're going to do here is just to pick apart then the contributions to the overall free energy change as you go from the unfolded state here to the folded state, the double helix down here. And this overall change, delta G, that's what determines how favourable a process is. It's made of two components. Delta H is the enthalpy change, which is the one that you're most familiar with, and that's the one that's going to depend on the changes in the energies associated with the bonds formed in the unfolded state and the bonds formed in the double helix. And typically that means that a lot of the interactions formed between the DNA and water in the unfolded state get replaced by interactions within the double helix and also between water molecules, which are released from solvating the separate DNA molecules when they associate together to form this structure. But this term, T is just the temperature, but delta S is the entropy change. And we've seen that entropy is a measure of the dispersal of energy within a system. And that's important because everything that happens in the universe tends in the direction of spreading the energy out over more possible energy levels than it was before. And just because of statistics, we saw that that's what naturally tends to happen in all processes. So you can predict what will happen and what will not happen in the universe, because unless it leads to an overall increase in the dispersal of energy, it's just statistically so unlikely to happen that it won't. The point about delta S is that it only focuses on that question of dispersal of energy within the system. But what delta G does is that it basically quantifies the dispersal of energy in the universe as a whole. The idea being that if delta H is negative, it means that heat is released from the system into the surrounding universe, and it therefore increases the dispersal of energy in the wider universe. So combining these terms, we basically combine the dispersal of energy within the system and the dispersal of energy outside, and delta G quantifies the dispersal of energy in the universe as a whole. And the way we summarize this thing called the second law of thermodynamics, which says that has to be an increase in dispersal of energy, is to say, well, delta G must be negative. So that's the significance of this term here. The negative delta G means that under physiological conditions, the double helix is stable. And what we're going to do is just pick apart the various contributions which lead to that overall change. First then, we focused on the backbone of the DNA, which is highlighted as space-filling atoms in this representation. And what we saw is that one significant issue is that these groups, which are the phosphate groups, are all negatively charged. And so there is going to be repulsion between them. Within the chain itself, that's just a fact of life, that's always going to be there. But what we identified is that when the helix folds up, you're going to increase those repulsive forces, because particularly across here, which is called the minor groove of the DNA, you're bringing, for example, this phosphate group and this one into quite close proximity. And that's going to be repeated all the way around the structure. So there is a significant increase in the proximity of these groups that repel one another when the double helix assembles. And that's going to be unfavourable. So returning to our summary free energy diagram, now we can add our first term onto this. And it's an enthalpy term, because enthalpy is the term that relates to changes in energy. And what we're saying is that there is extra repulsion, and that's going to require energy to be put in. In other words, it's an endothermic change to make that increase in repulsion possible. So far we've gone in the wrong direction. We're saying that that disfavours formation of the double helix. There was another really important thing to do with the backbone, which we also then considered. We explored the idea that because the backbone of DNA is made of single bonds, it has a great deal of rotational freedom, and that allows it to sample a great many different arrangements in terms of its overall shape. And in terms of entropy, that represents a high entropy state, because all those different motional modes that you're seeing there, in which different atoms are not just rotating relative to one another but are actually moving through space to different positions, represents a lot of different ways of dispersing the energy, compared to the double helix, where that freedom is largely taken away. In the folded double helical structure, we have a much more fixed structure. It's not completely static. So here's a simulation where you can see that it can wiggle around a bit, because there's still some mobility in individual bonds, but it's essentially restricted to just gyrating slightly around that equilibrium structure. Those dynamics are thought to be very important for function, but from our point of view, they represent much more restricted freedom of movement than we had in the unfolded state. In terms of our free energy picture, what that represents is that in the unfolded state, you have a lot of entropy, both because the individual chains, as we saw, are able to sample a huge range of conformations, whereas in the double helix, although there's limited mobility, they're much, much more restricted. But also, of course, because the two chains here are able to move around independently of one another, whereas here they're locked together. And both of those features represent a much greater dispersal of energy over different levels in the unfolded state than you would have in the folded state. So what that corresponds to is an unfavourable entropy term. So if we look at where that takes us to, we had the repulsion between the phosphates, increasing the free energy, and now we've got this loss of entropy. So now we're in a state where folding into the double helix so far looks really unfavourable because we've got a big positive delta G. These terms are not meant to be shown to scale, by the way, but it's just to make the point that this is a positive term. So both of these backbone terms are saying, yeah, double helix doesn't look like a good plan. So we then have to turn to the base pairs in the middle and see what they could be doing, which might swing the equilibrium in the other direction. So looking at the actual structures of the bases which form the heart of the DNA double helix, we saw that all four of them are these flat aromatic ring structures, but that they have a great deal of hydrogen bonding potential because you have all these nitrogens and oxygens with their lone pairs which can participate in hydrogen bonds, and you have all these NHs which can also, of course, participate in hydrogen bonds. And we saw that within the double helix, some, though not all, of this hydrogen bonding potential is realized by formation of hydrogen bonds between base pairs from one strand and the other. So you get these famous Watson-Crick base pairs between guanines in one chain and cytosines in the other, forming an array of 3 hydrogen bonds, and then adenines and thymines pairing to form a pair of 2 hydrogen bonds. But when we thought it through, we realized that if the chains fall apart and form the unfolded state, then these groups won't lose out on hydrogen bonding. They will simply hydrogen bond to water instead. And so it's not at all clear that formation of these hydrogen bonds actually makes much contribution to the stability of the folded double helical structure, because all you're doing when you form them is replacing one set of hydrogen bonds with another. That's really not to say, though, that these hydrogen bonds are unimportant, because within the context of the double helix, you can see that these bases are necessarily rammed up face to face with one another. And so if those hydrogen bonds were not formed, what you would have would be individual NHs or carbonyls or nitrogens with hydrogen bonding potential unrealized, because they would have been taken away from the water by the folding into the double helix, so they'd have lost their hydrogen bonds to water, and they wouldn't then be replaced by alternative ones if you didn't have the complementarity across here. And that would be hugely unfavourable, because you'd be losing hydrogen bond energy, which is a significant term. So that would represent a big positive delta H contribution if those hydrogen bonds fail to form. So that is why you get complementarity. G has to be paired with C, A has to be paired with T in order to get stability. We also saw that if you relax the constraints of the double helical fold a bit, then alternative hydrogen bonding patterns become possible. So you can pair G with T or A with C. And so that led us to take the view that whatever else is driving the folding of the double helix must have a very specific preference for the particular double helical structure that you get, because we get this fidelity of hydrogen bonding where it always pairs up like this. And that can only happen if the particular double helix that we see is significantly more stable than alternative structures in which these alternative hydrogen bonding patterns might be formed. So on our free energy picture, we've had our backbone contributions from the phosphate repulsion and the loss of entropy, taking us to a very positive delta G so far, and we can now add in the contribution of formation of hydrogen bonds, which to a first approximation looks like it's going to be nothing. So we're basically staying on the same level because we're just replacing one set of hydrogen bonds with a different set. The important thing about that, though, is that even to break even at that point, we had to have the correct complementarity. In other words, we had to have G paired with C, A paired with T, and that's the crucial thing in terms of DNA's function. So although it's not helping us stabilize the double helix, it's very important for not destabilizing it even more. So then we looked at the other intermolecular forces that the bases form, and we saw that because they're aromatic, they are flat, and because they're flat, they can stack very effectively one on top of the other to form this very densely packed heart of the structure, which you can see running through there. So we looked in detail at the forces that could be formed there, and we saw that the structure does its best to maximize the contact surface area between the stacked bases. So for example, by introducing the propeller twist, which we saw between one base and another within the base pair. So if we then start to pick apart why this stacking up of the bases is favourable, we identified really two types of driving force. One is that the intermolecular forces between the stacked bases are likely to be significantly more favourable than the ones that would be formed between the bases and water if the double helix unfolded. And we identified those forces as being kind of just induced dipole-dipole forces, because the flat surfaces of the bases are in good part non-polar. So we know that when non-polar molecules are attracted to one another, the induced dipole-dipole force is the key mechanism for that. But it was complicated by the fact that these are aromatic structures, and the interactions between the pi electron systems are very important in the detail of why those interactions are so favourable. We looked at the behaviour of water molecules in liquid water and saw that compared to ice, these molecules have much less regularity of structure and they have significantly greater freedom of motion, so they're able to reorient, rotate round, form hydrogen bonds with different partner molecules. But at the same time, they still manage to make almost but not quite as many hydrogen bonds as they would do in ice. And what we thought about was what would be the effect of exposing this kind of liquid water to a non-polar surface. So when non-polar groups are introduced into water, the problem from the water molecule's point of view is that they can't form hydrogen bonds in the direction where the non-polar surface is. That doesn't, though, mean that they necessarily lose out on the number of hydrogen bonds they can form, because they're able to form these kind of structures. So this is a model of methane surrounded by water, but the same kind of thing would apply when the non-polar surface of DNA was exposed. And what the water is able to do is to form an ordered sort of cage structure around the non-polar surface, almost like a thin layer of ice surrounding that surface. And that ordering allows the water to maintain its sort of 3.5 hydrogen bonds per molecule without therefore any significant difference in enthalpy compared to the liquid state, but it does that at the expense of having to be much more ordered than it would be in normal liquid water. So it doesn't have that, the molecules don't have that same freedom of motion that they would in the liquid state. So the consequence is that exposure of nonpolar surfaces to water decreases the entropy of the water. So in terms of our folding equilibrium, we have this situation that in the unfolded state, where the chains are separate, all these bases are now highly exposed to water, and the water surrounding them is, at least around physiological temperatures, going to be in this low entropy, highly ordered state, which it needs to adopt in order not to lose out on hydrogen bonding. When the double helix assembles and we move over to this state, a large proportion of the non-polar surface of the bases gets hidden in the core of the structure where the bases stack on top of one another, and so that highly ordered water is now released out into the bulk solvent, where it can become significantly more disordered. So what that's going to give is a significant increase in entropy of the water when you move into the folded structure. So returning then to our free energy summary, this is where we'd got to so far, having thought about the unfavourable contributions that folding the backbone makes to the stability of the structure, and the effectively break-even contribution that the base pair hydrogen bonding makes. We've now identified the stacking of the base pairs in the core of the structure as being a favourable thing, which will help to now drive the folding of the double helix. And we saw that that was really for two reasons. So first of all, there was this enthalpy contribution from the fact that the base pair stacking makes very effective intermolecular forces as a result of the very close contact of those planar surfaces. So that has helped to take the free energy right down, and then added to that, we have this favourable entropy term. So an increase in entropy giving us a negative contribution to delta G, resulting from the fact that we've released the highly ordered water that would be surrounding the bases in the unfolded state when they stack together, and that increases the dispersal of energy in the system. That's called the hydrophobic effect, and that's also a significant driving force for the folding. Thinking about the entropy of the water made us realise that there was also going to be another contribution to the thermodynamics of folding, resulting from the water molecules that fill up the grooves in the DNA surface. And especially in the minor groove, the narrower one here, we saw that the water had to be very ordered into essentially forming this spine that runs all the way around the minor groove in order to meet the hydrogen bonding potential of the groups still exposed to the surface after the double helix has assembled. And so that's going to make a significant unfavourable contribution to the stability of the folded state, because again that's decreasing the entropy of the water, not as a result this time of exposure to the non-polar surfaces, but of the restrictions on the interactions formed with polar groups because of the limited space within the grooves. From the point of view of our free energy diagram, this ordering of water within the grooves when the helix assembles is going to make another entropy contribution, and this time it's an unfavourable one. So this is going to take the free energy up in the positive direction again a little bit. But it's not enough to make the overall process unfavourable. So you can see, once we add together all of these contributions, what we end up with is an overall negative delta G, and that's what makes the structure stable. And it's not all that stable. The delta G is relatively small, and of course that's important because we don't want the DNA to be solid as a rock. It has to be unfoldable because that's necessary for the sequences of the genes to be read and transcribed and translated into proteins, and it's also necessary for when the DNA gets copied, for when cells divide. So ending up with this structure which is marginally stable is actually quite important. And we can see that arriving at that point is not as a result of any single term. It's a very complicated picture in terms of lots of contributions from different types of chemical effect. And that's basically the end of the story.